Is okay?
Yes, sir.
So what we are actually doing in this workshop, we are trying to understand so-called structure property relationship using either atomistic or electronic structure methods. And our fundamental aim is to understand and provide a foundation about the atomic interactions in a given solid 3D, 2D, 1D, or 0D. And once we understand the interactions, we can easily predict the electronic, optical, or mechanical response of these materials. So in my group, we have been doing a lot of work. I started my career as a crystals, the bulk materials, mostly inorganic, and then moved to clusters, nanostructures, 2D materials. Although I'll not talk about it, but my current interest is, and we call nanobio word, or the buzzword is we call biomolecular electronics. But next time, if Dr. Singh is going to organize another workshop, I'll talk more about nanobio. So this outline of the talk is just brief discussion about what the nanomaterials or low-dimensional materials are. Then you may have, again, heard few talks about density function theory this morning in the workshop, which is the basis for doing a simulation. And I'll pick up a few examples depending on the time. The DNA sequencing, sensing, and what we call device characteristics. So we have to be really thankful to experimentalists because their work, we had sort of significant advances in imaging techniques. So what we look into sort of a surface topography at an atomic level. So we had two types of tools available. One is the atomic force microscopy, where we have magnification in 3D, which sort of gives us a high contrast, and we can look into the physical properties. On the other hand, we have called scanning electron microscopy, SEM, which provides a magnification in 2D, which sort of gives you high depth of field. And it sort of provides information about the chemical composition of the surface. So here is one AFM image where you can easily see the gold clusters of about, say, few tenths or few hundreds of atoms with the nanometer size. So can I request for the participants, if you have any questions, please raise your hand or text it at the same time, because I really welcome the questions during the talk rather than after the talk. So here, this is a sort of a nice way of describing the relationship between simulation and what we have called imaging techniques. So you have on the y-axis the time scale, and you have x-axis length scale. And if you look at it on the corner, sort of a right-hand corner or left-hand corner, if you're viewing, we have so-called optical microscopy, and we have the tools coming from, we call the continuum theories or sort of a classical models. But as we go down on towards the nanometer length scale or femtosecond, picosecond sort of a time range, what you can see here, we have the high resolution TEM or SEM, but also we have the tools of quantum mechanics or sort of molecular dynamics in that sense. So these are based on DFT. So I like this slide is given by Professor Pandey from Delhi. Then what we have is look at the graphite pencil. And you can see that the tip of the pencil is the original size. As you sort of zoom in about 300 times using an optical microscope, you can begin to see sort of the surface, the tip of this pencil. And if you further zoom in,
So mute, yeah, some noise is there.
Is there any question or should I keep going? Okay. And then if you zoom in 90,000 times using electron microscope, you can really the topology of this graphite surface. And if you go further down about 27 million times using STM, you can easily see the hexagon rings made by carbon atoms on this tip of the graphite. So you can here easily see the advancement in imaging techniques has provided really precise details of surface topography at atomic level. If you look into this now, a cartoon, and what we have here is the cube. And if you look at the cube, take it centimeter by centimeter by centimeter. So what we see is the total surface area is 6 centimeters square because cube has 6 sides.
Gaurav, mute yourself. Gaurav, mute all.
Can you? Yeah, agreed. Thank you. And then if you go to a millimeter cube size, you can see going from centimeter to millimeter. Should I continue?
Yes, sir. Yes, sir.
Good. So going from centimeter to say millimeter cube, what we have is we gained in surface area about an order of magnitude. But look at here from millimeter cube to if you go to a nanometer size cubes, and what we have is a tremendous advantage in the surface area. So all these low dimensional materials, what we gain is the properties, spatially we can exploit this large increase in surface area to look into it. And that's the strength of these nanomaterials, is providing us a significant increase in area for reactions to take place. So this is, again, as I use this slide to explain, especially the participants, the classification of these nanomaterials. That's based on what we call is spatial confinement of electron hole pairs within a material. So if we confine these electron hole pair, we call excitons, into the 3 dimensions, what we have called 3D thing. If you go 2D in XY direction planar, we have a quantum well. If we have axial direction, we have a sort of quantum wire. And if we have a confined from all directions, we have a quantum dot, like we have a C60 or other quantum dots, CDS and so on. So what happens in we have quantum confinement? So what it does, it basically modifies a density of states. And as you may know, is in our electronic structure calculations, these are really the properties, the density of states, what we calculate, defines the response of the material. What does it do? It basically provides information about the number of electron states which are occupied in a given system per unit volume, per unit energy. So if you look at it, this energy distribution of these states which are available to us are for electrons or holes. For a bulk, you have density of states versus energy. It's very, very broad distribution. As you move from 3D to 2D, you can see again is a little more sort of discrete, sort of a localization of the states. For 1D, wire is a very, very narrow distribution. And you can see the quantum dot more or less behaves like a molecule. We have really localized electronic states. So if someone asks, the question is, what exactly is being sort of modified as we confine our carriers from 3D, 2D, 1D, or 0D, basically is what they are doing is modifying the energy distribution of these carriers, what we call density of states. So let's move to the theory. As what we use here is so-called density function theory. So what it does is the program. What we have is we start with sort of initial guess. So we have, if you look at, say, MgO, sort of a crystal, or even a graphene, 2D material. So we start the initial gas of the carbon atoms. And these density of carbon atoms is available, as has been calculated with a highly accurate sort of a quantum chemical calculations. And once we have it, we look into, we call the effective potential. We calculate the effective potential, and then we solve these Kohn-Sham equation. So this is your Hamiltonian. If you remember, H psi is equal to E psi, where H, this is the kinetic energy term, and this is the effective potential acting on the wave function, gives the eigenvalue and the wave function. And from once this we solve it, we evaluate the electron density and total energy of the system, where total energy, as you, if you remember, is the expectation value of a Hamiltonian with respect to the wave function. And we have some sort of a criteria where we say if the calculation or the total energy is within that criteria, which is very, very sort of small, then we say calculation is converged. If not, then it goes to the cycle. This is called self-consistent field approach to solve a Kohn-Sham equation. And once you have the energy and the sort of a density, then you can easily calculate the other properties. So the trick of the trade is what we have is we calculate energy surface. So you are calculating the total in the system by varying the electron density. And once you have the minimum in energy, what we call is a ground state of the given system. So let me give an example about calculating so-called these rare earth elements interacting with the benzene. So here is the benzene ring, and this is pick up a cerium, and then you go through a series of rare earth, which valence configuration, and you look at the equilibrium configuration, what the distance is, what the binding energy is, and the other properties are. But I focus on a cerium is, you see, the binding energy for a given system is about 7.2 eV. But if I change the system, again is within about 7.18 eV. So there is some change in the binding energy depending on the modeling element we use in these calculations. So what is coming out is you can see is we have is so-called numerical errors, and we have also called systematic errors. That's I would like you to get exposed to it coming out. So numerical error is, in a sense, what we sort of make approximation to solve this Kohn-Sham equation, which I talked about here. This is our Kohn-Sham equation. So in order to solve it, we have to make the sort of transformation to matrix equation, and that sort of introduces a numerical errors. So what we call is we have the finite basis set, which I think Dr. Verma is going to talk about, or Dr. Ashok. Then we have, again, a finite K-point mesh, and we have called finite integration grid. So these sort of introduces the sort of numerical errors. So we can say the bond energy approximately is the order of a milli-electron volt per atom, and we can calculate the bond distances or the lattice constants very precisely up to 0.001 angstrom. But we have also, we call systematic errors. So because of the limitation of theoretical model itself, if you go back to your quantum mechanics 101, we start with a many electron system. Then we have the Born-Oppenheimer approximation. We freeze the nuclei and calculate only the electronic part of the Hamiltonian. And then further in this electronic part, what we have is we use this approximation of exchange correlation functional form. And depending on this exchange correlation functional form, which is semi-empirical, what we have is a sort of a error coming into the approximation. And then we also have is so-called the self-interaction error, because the way we define it, this electron interacting with other electrons also induces the term the interacting with itself. And that sort of gives you the chemical accuracy of about a tenth of eV for energies, and one has to be very, very careful about it. So what is numerical precision is coming out, as I said earlier, is incomplete description of the orbitals, which you use to define a electronic density, because the basis set is a mathematical functions, and always we do not have so-called the complete basis set. We always use approximation to do it. And the reason being here is we have a periodic system, especially low dimensional material is in a 2D plane. And what, if you remember, you have the block theorem, where sort of brings in the periodic k vector, e to the power ikr, and the total energy then becomes a sum of these eigenvalues. So in a sense, we have a continuous space in the Brillouin zone where one should be using a infinite number of k points. But in practice, we use a finite set of k points, which are sort of gives you the error in these calculations. And finally, is what we have is when we evaluate exchange coefficient functional form, you can see is again is the weight factor Wi and a set of Ri, which are the discrete points in real space, but should be using a continuous space. But that's fine. That's one needs to use these approximations to calculate the total energy of the system. So you may have gone through the what we call variety of sort of we call Jacob's ladder. Is a very nice paper, and back in 2022 came out. So what you have is you start with the Hartree approximation. This is a quantum mechanics 101, where you do not have the exchange coefficient part. Simply you have the Hartree's repulsion, and you further move is the local density. So we have the density R, which is again is a homogeneous electron and gas density. You calculate in LD approximation. You move to GGA, and then we have is a density, but we also take the gradient of the density. And then you have so-called meta-GGA, hybrid, and so on, where you have again further refinement in approximating the exchange and coefficient part. Then I talked about self-interaction error. And as you know, is DFT is always systematically underestimates the band gap. And the reason being is here is electron interacts with itself, which sort of causes what we call is over delocalization of electron density. So what happens is what it does is if you have the valence and conduction band, this self-interaction error sort of promotes or provides upward shift. The band and your calculated gap is much smaller than the experimental gap. And this gives you an idea about if you took the computed band gap with the experimental band gap. So these are the calculated one are really underestimated by at least 42 to 50%. And one should be aware of this sort of the weakness or the shortcoming of the DFT method. Finally, what we have is introduction of what we call is DFT. Does not take account sort of accurately dispersive forces, or called Van der Waals forces. And the reason being is the exchange coefficient function form is based on these local or semi-local properties of electron density. So you can see here is for a graphite as a 3D material, if I calculate the binding energy per atom, my LD approximation gives about 17 milli-electron volt. If I include this Van der Waals term to take about the dispersive forces, which are sort of dominant in a system like graphite, and we have about 45 milli-electron volt, and experimental is 52. And interestingly, if you can see, LDA gives bound, but GGA is not bound at all. So again, is what I'm trying to give is, yes, DFT is a very powerful tool, but one should be aware of the so-called systematic or chemical errors before you go on predicting the properties. So why are we doing it? So our idea is we have to provide the results of such accuracy that we can predict the physical and chemical properties of materials. That's our goal is. And this capacity is desired on two grounds. First, what we do is we take the system, which we have the experimentally known properties. So we calculate those properties and compare with the experiments. What it does, it gives us a confidence about how good our model elements are, which are the basis set, would be plane wave or the Gaussian basis set, the K-point grid, the integration grid, and then we have the exchange duration functional form. And once we have it, when we sort of verify these model elements, then we can go on and predict the properties. And there are two ways. Experimentalists, they have these properties, they observe it, but they don't know what is causing it. So we can help them to understand those observed ones, or if they are inaccessible to experiment, can go on predict those properties. So let me give an example. Is nanoparticles in environment related to it. So one of my colleagues, Dr. Hussain, Sabir Hussain, came to me. He's experimentalist at the Air Force lab. He said, Ravi, this is the result I have found. Can you explain? Can you find a sort of a reason to do it? So what he has done, if you recall, he looked into dopamine. So dopamine is a neurotransmitter we all have in our brain. And if you become happy, the level of dopamine goes high. So he's monitoring the level of dopamine in so-called these PC12 cells. So what he finds, if you look at it here, this is the cell count, and this is the different nanoparticles. And if you look at the manganese, either in a pure manganese form or in manganese acetate, what he finds is as he increases the concentration, level of dopamine goes down. But if he does the silver more or less is not affected. There is some decrease, but not as much as he has found for manganese. So his observation was manganese depletes the dopamine level in these PC12 cells. So we came up, as you know, we had to start with a hypothesis and sort of make a plan for doing the simulation. So our question was, we would like to address is why do these particles deplete level of dopamine or dopamine? So what is happening here? So in a sense, we have to get down to at atomic level. We are looking at the interaction of these particles with the dopamine molecule. So what we need to understand the interaction is, in a sense, we have to calculate these called energy surface, where Mn13, I picked up the Mn13 sort of a particle, 13 atom manganese, interact with the dopamine. And then silver 13 is interact with dopamine. Then what we did is we realized is if you look at the electronic configuration, manganese is 3d5, 4s2. Silver is 3d10, 4s1. So we thought is we had to have a reference case. If you have a d electron interact with the dopamine, s electron interact with the dopamine, why not pick up a p electron system? So that sort of gives you s, p, d electron interacting with the dopamine as a nice sort of a reference to compare with. And therefore we picked up is Al13 to calculate. So for this energy surface calculation, what it does, it gives the binding energy. And once we have the binding energy, we know what kind of interaction strength of these metallic particles having with dopamine. So this is a cartoon sort of thing. What we are looking at is, as you know, dopamine has a sort of a different sites. So these particles can come from OH side or from top side or coming from amide side. So we're looking at the energies surfaces on all sort of different sides. So we calculate total energy versus the distance, and this case is the nitrogen side is interacting. So what we have is the total energy. So as you know, is a far away. There's no interaction. It comes down, electrostatic interaction, and at certain level reaches and then goes up because we have the Pauli's repulsion between electrons. So all the curves show this kind of behavior. And from here we find is the top side is the most stable side because it has the lowest energy or the highest binding energy. So from there, we calculate the manganese 13, which is a D5 electron, has the binding energy of about 1.22 electron volts. Silver 13 has an order of magnitude less, which is the S type, about 0.12, and this Al13 is not bound at all, which is P1. So this gives a nice sort of a contrasting behavior of these sp and d electron system interacting with the dopamine. So we thought if this is the case, why not we look into the charge density plots? So the assumption is if is this tightly bound or is it strongly bound, then definitely is going to have a large overlap of electron density coming out of the metallic nanoparticle, and you have the dopamine, this thing. And indeed, that's the case is Mn13, is you have the large overlap compared to silver, and here there's hardly any overlap at all, which is not a bound system. So it comes down to the forming of a bioconjugated complex is simply overlap of electron density, and what we have is orbital hybridization. And if you coming from physics, I talked to my chemist friend, Dr. Karna. He saw these results. He said, wow, it does make a sense. The reason being is you have the dopamine, which is a carbon rings. It has a pz electron sort of forming a pi electron cloud. So this pi electron cloud interacts with the pd interactions very, very strongly compared to ps or pp hybridization. So in a sense, what it comes down to the result is the calculation is sort of reaffirming or confirming the experimental observation that manganese 13 particle, they strongly bind with the dopamine due to this PD hybridization with the electronic cloud of a benzene. So they have a really high interaction strength, and you expect the manganese level to go down because they form a very strong bounded complex of dopamine compared to either silver or aluminum. Any questions so far? Dr. Singh?
Presently, no, sir.
No? Okay, good. So let's move to the second thing is I call is how good the models are. So on the left-hand side, what you have is the experimental cross-sectional TM image. So what you have is this is substrate, silicon carbide. Then you have the so-called the gallium nitride, the monolayer. You can see this is the bilayer case here coming up. And then you have a graphene bilayer. So you can see really is the scale you can see here, the 30 angstrom. You can pinpoint the atomic interface formed by this gallium nitride and the graphene systems. At the same time, this is one of the examples from our calculations, is here what we have is a carbon nanotube, you can see, and then here is one of the DNA molecule called guanine. So what happens is carbon nanotube, again, carbon atoms, they have pz orbital forming this called pi electronic cloud. And same thing is with the benzene ring in guanine forming a pi electronic cloud. And you can see we can easily simulate such a weak interaction between this pi electronic cloud of guanine and carbon nanotubes. So in terms of the energies, if you go back and look into it, once we calculate what we call the energy surface, you can see is the accuracy of energy surface is about 4/10 of electron volt or 4 electron volt, which is pretty accurate in energetic sense. So what in our group, we had done some calculations to look into how does this carbon nanotube interacts with a metallic nanotube. Sorry, the guanine or the DNA molecule, what we have here on top, interacts with a metallic carbon nanotube or semiconducting boron nitride nanotube. The reason being here, what we have is this is a metallic, so it's a purely van der Waals type of interaction. And being a boron nitrogen, because boron and nitrogen, they have a charge transfer. So what we call is a semi-ionic bonding. And you can see the calculations distinctly differentiate these two types of bonding, which is what you call is the dipole-dipole interaction between pi electronic cloud of guanine and a carbon nanotube. And once you have the semi-ionic bonding of a BN and T, in this sense, you can see this sort of forms a covalent type of interaction we call polar covalent interaction between boron nitride and guanine. So the examples I've given you can see is both in terms of the energies and electron density, the description of electron density. These siesta or a DFT-like calculations can provide very accurately. So let me pick up an example. This is a few years ago we did, is can we sort of help out in DNA sequencing? So the idea came is we saw one paper that DNA wrapped CNT can act as a biosensor. And you can see here is this is they have fabricated the device. You have source, drain. There's a carbon nanotube wrapped with the DNA, and you have the gate voltage. So once you for the given source and drain voltage, what you're doing, you're applying the variation in the gate field. So what you have is x-axis is the gate field, positive and negative, and then you have the conductance. So what you see here, the black curve is a bare boron nitrogen, bare carbon nanotube. And once you have the DNA wrapped, we call the probe is red. It's a distinctly different signal. So this DNA wrapped carbon nanotube, when sort of acts as with any the sensing molecule, what you have is a green curve, which is a hybrid. So the paper sort of shown that a DNA wrapped CNT can identify, depending on the shift in the conductance, the molecule is being sensed. So the question we asked is, if this is fine, they have fabricated the device, what kind of interaction they are having at the atomic level? So we started with the approximation because, as you know, carbon nanotube sort of have different sizes and different types. So we thought, let's pick up a very small diameter carbon nanotube. It's a one end. And suppose we take the very large diameter carbon nanotube, which one can be approximated by a graphene surface. So what we have is these DNA bases interacting with a small diameter, and a large diameter gives two ends of the interaction strength. DNA, this is again is a quick update or 101 thing is what we have is a double strand DNA connected with these called sugar phosphate backbones, and we have the base pairs, which is our adenine, thymine, cytosine, and guanine. I can see is nothing but the sort of a benzene rings with a different functional groups coming out. So you're looking at is I said our trick of the trade is sort of determine the energy surface very, very accurately. So we have the periodic supercell of, say, graphene or carbon nanotube, and we are bringing in these the nucleobases to calculate what is the minimum total energy of that complex or grounds to the complex. And there are sort of different ways of, because this is a complex system, you go one by one to look at so-called selective dynamics, grid scan, root scan, or full relaxation. And one of the examples is coming out is really you want to see, you have first optimize, I call the z-axis distance, and then you rotate the molecule to look at the energy surface, is which one sort of provides a minimum energy, and then you do the full optimization. So this sort of a energy surface comes out that this is the maximum, this is the minimum, and you can see the difference is about 0.15 eV, which we are looking at is we are sort of picking up the surface of this carbon nanotube and see how does my DNA nucleobases interact. Once you have it, then what you do, you calculate the binding energy. So binding energy is going to be the complex total energy, and then you have the constituents energies of the system. Take the difference, and what you find is what we started with the approximation is we have small diameter, and we have very large diameter graphene. And hypothesis was is this is going to give us at two ends about the binding energy. And you can see the follows the pattern, which is similar to each other. We have the bases, adenine, cytosine, guanine, thymine, and uracil. We picked up the uracil being an RNA compound, and this is the strength of the energy. So this sort of comes out as your carbon nanotube, and this is your graphene. And you find is indeed the guanine interacts very, very strongly. Does not matter is a small diameter or large diameter carbon nanotube. Adenine cytosine has a different variation because of the curvature effect, but thymine and uracil sort of again have different responses. So what we found is these base molecules interact differently with carbon nanostructures. Could be a nanotube or say a graphene. So that's our observation, that's our prediction. So the idea came in is can we use this distinct sort of interaction strength of these carbon nanostructures to look at the current passing through the nanostructure? Because this is nothing but what we call is a DNA sequencing. In practice, what it happens is they use is a gel phoresis sort of approach, where they go by sort of one by one, but we have the challenges in this conventional approach is a limitation of the length and the pre-processing steps and then post-processing steps. So it takes time, and of course it's still available. But our idea is let's go back and see can we use this distinct responses by looking at the sequencing through the transport. So this is fine. So I asked the student to look into it. And interestingly, we found is the literature, people have studied this sort of approach previously. So what here they're looking at is they have, say, graphene, a carbon graphene sheet, and they have the whole. And as you know, so double-strand DNA sort of split into single-strand DNA. And being it has a charge, if you let electric field, you can pull through the DNA through the hole. So that's they measure the so-called the transference current going through as the DNA and nucleobases passes through it. So you can see here is a normalized current. And so you have G, C, T, and they have distinct responses. The other calculations sort of predict the same thing. But what you have is the responses are different, but a lot of noise is there. Signal is not that clean. So what we realized is yes, people have done it, but the approach is so far is not sensitive enough, and because it sort of gives a large fluctuation because of this current is not that stable. So our idea was then come out is use these nanopores, nucleobases, and can we use as sort of a probe to stabilize the passing DNA interaction. So what we have is previously they had these pores with these other contacts without this probe molecule, and simply a DNA, single-strand DNA is passing through it. So what we suggested, let's add this probe to our nanopore contact and see if this interaction between the probe and the DNA can stabilize the current. So this is the model system. You have the contacts, the gold. That's our probe, and this is our target passing through it. So this is what we use is so-called the semi-empirical non-nucleum green function approach. So from DFT, what we have is, this is the way. I have the cartoon. You can see what we are calculating the current in terms of these functions, this Green's function approach. So what it does is, see if it works. So I'm plotting current versus voltage, and you can see here this is the left contact, and this is the right contact. And what we have the eigenvalue of the active system or quantum region, which in this case is our DNA passing through the nanopore, the energy level. So as we apply the voltage, it doesn't work that way. So what happens is this level sort of shifts upward and this sort of downwards, and one sort of it matches the current flows. That's the idea of this sort of a source drain two-terminal device model. So you can see here in the simulation, so what we are trying to do is we are calculating the current as sort of it passes through by varying this probe molecule, which is attached to our contacts. And from there, what you do is you calculate the energy surface and find, calculate the current also from there. That if there is a order of magnitude difference in current, so we start with all the four, adenine, cytosine, guanine, thymine, and there's a high current. We have the one sort of a loop, and then low current, we have the one loop while at the bias about 0.1. And then we go further, we increase the bias at different fashion. You can see we can distinguish between C and T, and at higher bias between G and A. So given on this probe, what we have, we can sort of define or distinguish from A, C, G, T.
Excuse me, sir. Can I ask one question, sir?
Please, please, please.
Sir, what is the name of the probe molecule that you have used here?
That's a good point. That's a good point. And that's what you can see in the next slide. What we have is, depending on the interaction strength, which I can show you later. So what we have, the probe molecule, we have ACGT. And as you know, the AT is really a favorable and GC is favorable. So as you go through, you find the signal is coming out because of the conjugation of AT. And as you go down, you can different pairs and a different sort of a current. So this comes down to this interaction of a given probe. Here we have the C probe. And you can see the C probe is going to interact with a higher affinity with the guanine, which is coming through the channel. So we have used all the four probes, and depending on it, the interaction sort of comes out, giving signal in a different channels. Does this give you your answer to your question?
Sir, it's interesting that guanine is interacting with guanine.
In this case, but you can see here is the channel, the signal is going to be weak or strong. If you see here, it can be low, can be high. So between the difference in current, you can distinguish the which is going to be there. And this is simply a cartoon. The maximum is coming out from G to C range, and it goes through it.
Yes, sir. Thank
And
you.
the reason being here, this sort of gives you a reason. Guanine is going to interact guanine, but the question is going to be very, very weak interaction. Guanine, when it comes to the cytosine, GC pair is going to be very, very high interaction. So what is happening here, you can see the probe and the different DNA. We are looking at the subtle variation in electrostatic potential, depending on the target or probe molecule. So this subtle variation, you can see mostly is the same. But during this interaction, this small variation gives you the low current or high current. And we sort of exploit this difference in low or high current to have the different channels. So low current GG interaction gives you at least some magnitude, which is say, aha, this is a weak or low current is going to be definitely GG or AA or CC in that sense. But GC is going to be very, very high interaction, high current. Is that okay, Dr. Vishwas?
Yes, sir. Yes, I get the answer, sir.
Good.
Thank you, sir.
So is it simple as I understand is you have the hydrogen interaction depending on the magnitude. It sort of manifests into the electric current, and then we exploit it. So how many minutes do I have? Another 10 minutes?
Yes, sir.
Yeah. So what I do, I'll give you just change the topic here. And just show you is what are the limitations of these nanomaterials when we are trying to make the electronic devices. So what we have is a field effect transistor, and this is I call it device physics 101. So what you have is a source and drain. You have channel, and these are the insulating materials. And what your idea or our idea is, by sort of varying the gate field, you can have the different source drain sort of a relationship or different current at a 1G, 2G, 3G, depending on this the magnitude of the gate field. So what it does require is the channel material should be semiconducting. It has to have a gap. It doesn't matter what the magnitude is, but the gap is a necessary requirement. So you heard about the graphene, and as you know, graphene is a zero gap material, but it has a very, very sort of high in plane electron mobility. But being a zero gap material, simply we cannot use graphene as a field effect transistor channel material. So what people have done is because we have a tunable gap, they have sort of used a graphene bilayer. So what is the hypothesis is you have the graphene bilayers, monolayer graphene 1, graphene 2, you apply the electric field. So electric field in the bottom layer, these carbon atoms experience a different electric field compared to the top layer. And this a difference in electric field experience induces, we call the inhomogeneous charge distribution in the carbon lattice. So what it does is this inhomogeneous charge distribution sort of opens up the gap. And then you can use a bilayer graphene as a field effect transistor channel material. Monolayer, we cannot use it. So here is the experimentals have used a bilayer applying the electric field. So you can see, so you have to have, besides the gate field, you have to also apply the electric field. So this gives added complexity in fabricating a device. So when we saw this paper, we sort of thought about it. And we thought is there any way to induce this inhomogeneity in the carbon lattice electron density distribution? So we came up the idea is we have the graphene. Why not we make a heterostructure with a graphene with a boron nitride monolayer? Advantage is graphene and boron nitride. Graphene is column 4, boron column 3, nitrogen column 5. So they are structurally similar. They have a very small lattice mismatch. But the ionic bonding is different. The carbon is purely covalent, and the boron nitride we call partially ionic. So our expectation is if one of the carbon atoms, we make a heterostructure, sees a boron, and other carbon atom sees a nitrogen. So this boron nitrogen sort of electron density being different is going to induce a inhomogeneous charge distribution in the carbon lattice. And that should open up the gap in the heterostructure. So if you look at the left is a graphene. Then you can see is the Dirac cones nicely comes out, and it's a 0 at a K point. We look at the boron nitride monolayer, and you can see is a nice sort of gap opens up, being a semi-ionic. It's about 3 electron volt. And if we take the reference case of a boron nitride bilayer, and you can see monolayer, bilayer sort of mimics a similar band structure. And if you look at the heterostructure, what we would like to achieve to opening up the gap at a K point, so we have a graphene boron nitride heterostructure. And you can see here is this more or less similar to graphene. But if you zoom in at a K point, what if we find is indeed there is a gap opening, which is about a few millielectron volt. Now, the question I asked the student who was doing the simulation, is this real? It could be a sort of artifact of the calculation. So how to test it out? So our hypothesis is this interaction between graphene and boron nitride induces a gap, but this is the ground state of the heterostructure. What happens if we increase this overlap of electron density by decreasing the distances between these two monolayers? Would it increase the band gap? So what we do is this is our equilibrium, and we are looking at the change in the band gap by reducing the distances between these two monolayers. And you find indeed that a gap really opens up by about 10% reduction in the interlayer distance is about 100%. If you do the same experiment with the boron nitride bilayer, nothing happens. So indeed, is what it does is we can induce the inhomogeneity in the carbon lattice of graphene by bringing in a structurally similar but chemically dissimilar sort of a monolayer, which sort of the interaction opens up the gap, and one can use a bilayer graphene boron nitride system in field effect transistors as channel materials. Any questions so far? So let me wrap it up here. I think we are over time, but do it. So hopefully I have provided you an idea about indeed simulation in theory is a powerful tool. It does provide a fundamental understanding and sort of a foundation of these interactions between atoms or molecules. And once we understand or once we simulate these interactions in a more reliable or accurate way, we can easily sort of calculate electronic, optical, or mechanical response of these materials for energy and health-related applications. So let me thank, as we know, we had to grateful to our gurus, students, postdocs, and collaborating colleagues over the years. Let me thank Dr. Singh for inviting me to give a talk, and of course Mohan Verma, Keshav, Neha, Ashok, and other team of the members who are organizing this workshop. Thank you very much.
Thank you, sir. Sir, one request, sir.
Please.
Actually, this lecture is being recorded in your system, sir.
Okay.
So
So I
once
have
it is
to send it to you.
recorded, send it to Mohan, sir.
Sure, I'll send it to you.
Yes, sir.
And I can share also these slides also if you want.
Yes, yes, sir. We need.
That's not a problem. This is an open thing. And what I have a lot of material, so let me clean up and I'll send it to you. So you can
We are
distribute to
open for
participants.
questions if participants they want to ask any question. Anything.
Thank you very much, sir, for your illuminating lecture, sir. I have learned a lot from you as well as Dr. Mohan, sir. Sir, I have one question. During computational material science research, sir, as graphene has zero band gap, but we do find some kind of band gap, and
Yes.
how we can get zero band gap, sir?
No. The question is, Dr. Schwanz, is the DFT provides you underestimates the band gap. Okay. So suppose even you have a gap which is very, very small, but experimentally it has going to be a finite gap. So what we have is, I don't know, Dr. Verma is going to teach you, but we have the called advanced exchange correlation functional form.
Yes, sir.
So it provides a sort of a accurate band gaps, but cost is high. So
Yes.
what our idea is just use what is
Sir,
given to you, which is easily
muted, sir.
one can do it.
Sir,
And then from there,
have
you can
muted.
do these
Your voice is
accurate
not
calculations. Sorry?
नहीं, Keshav, clear है. Yeah. Voice is okay.
Yeah. So Dr. Schwartz is there is always a tricks to use it. And if but again, one should be aware of it. The band gap is underestimated.
Yes,
So what
sir.
we are looking at the trend in that sense.
Okay, sir. Okay. Thank you. Thank you very much.
Sajal is a new member of our team, sir. He's taking
Good.
one hands-on session in this program. And also we have
Namaste,
Dr. Imran is also good in.
So
Yeah.
good evening, sir. My question will be a little bit lengthy, so
Sure, go
that
ahead.
you understand the scenario before the question. So basically, I am an experimental guy. I mean, I did experiments, and where I studied the nucleation growth of nanoparticle at Synchrotron at Germany, Frederick Alexander University.
Very good.
And believe me, we spent a lot of time by by writing a proposal and then submit and wait for 6 months and then get appointment at the synchrotron by
Yeah.
measuring the distance of citrate molecule with the surface of at the surface of nanoparticle, because my nanoparticle were zinc oxide and we decorated them with the citrate molecule. Because the citrate molecule give them extra stability. Approximately, we did a lot of experiment and we found that citrate kept zinc oxide nanoparticle can stay stable up to 2 months. And the size of nanoparticle were just like 3 nanometer. They are in quantum dots region, yeah. So after that, it was for the cross verification whether I can do the same thing in DFT. So I joined Professor Verma group and learn a lot of things. But you know, the computational cost actually restrict us to the zinc oxide surface, not exact nanoparticle. So then I place the citrate molecule. Believe me, I published that paper 2 months ago. So
Very good. Congratulations.
yeah, thank you. And believe me, I received exactly the same distances between a zinc and the carboxylic acid group of oxygens, where we spent 1 year for about this distances. So I was
So
happy. My one question is here whether the
please.
approach is fine, because I consider the zinc oxide surface while there at synchrotron, basically we were dealing with the nanoparticle of 3 nanometer.
I think I presume yes, because as you know, 3 nanometer nanoparticle means you are talking about 1,000 of atoms.
Yes.
If you look at the 3 angstrom distance between zinc and oxygen, about 2 angstrom, and again you cube, so 0.2. So at least to me is 1,000 atoms. And what you are doing is you're looking at assuming that these nanoparticles provide or exist in a bulk phase, crystalline phase, which I presume it should.
Yeah.
Okay, so if the beam is coming in, and as you know, is a very, very narrow beam, is going to interact only the fragment of nanoparticle. And then
Yes.
definitely can be represented easily by the surface. Because ultimately we are looking at the local phenomenon, not the delocalized, is the localized. So your assumption, approximation, treating nanoparticle surface as a zinc surface, zinc oxide surface, I think is the correct one. That's why you got the distance. Correct.
Oh, okay. Thank you. So I was just curious about that, whether
No,
I
it's,
was in the right direction.
always remember we have to, you are experimentalist, and I always thank experimentalists to lead the way in doing nanomaterials research. But the theory sort of comes in to help you, assist you in
Yes,
understanding the result.
basically, sir, we are performing the atomic pair distribution function analysis,
Agreed.
where we see the exact localized distances between the atom.
Yeah.
So my supervisor basically belongs to the very accurate distances, because XRD people are synchrotron people.
Yeah.
They are only interested in structure.
Yeah.
So when I represented this result to him, he says, "Wow, we spent 1 year to get these distances, and you get this in just like in 1 month." So he was not
So
believing that.
no, so the DFT, as Dr. Vishwas also talked about, is very good in its structural properties. So the distances you are going to get is very accurate, 2.001 angstrom, 1,000 of angstrom. The gap wise, because what happens is the excited state kicks in, and that's why it's underestimated. So the structural properties, I can assure you, even with the LDA or GGA, you can get very accurately. Now, I think the key thing is see how does the electron density look like. That gives you added information about the bonding at the interface.
Yes, that was interesting. Yeah. So actually, I did everything. I mean, I check the bonding schemes, and we also performed the COPE analysis for that,
Yeah.
and the charge density differences, where the charge, how
Agreed.
the citrate molecule
Yeah.
interacted through, what are the active sites. But I mean, in experiment, we spent, believe me, more than 1 year. While in DFT, I perform just like in 1 month.
And that's the strength. You're right. Is coming out.
And the last question from my side is about some time when I put some vacuum on the surface to interact the molecule with, I get some straight line, bent lines. And I didn't get because I searched a lot of time about that, but I didn't get the exact answer for that. Why straight bent lines?
In what?
Just like last time, now I forgot the material, but last time I performed some calculation. I think so it was a heterostructure we simulated for, and the manuscript is already submitted. We calculated for VBIO4, and some regions shows the straight lines.
No, straight lines in what? Is a band
In
gap,
band,
dimensional
yeah,
states?
no, in band plot.
So I think that could be the reason is because you're not picking up the very, very high resolution we call is contour size. So contour size is going to be very, very high or very, very low. So you have to play with the contour size. So what happens is if you use the high, then basically you're zooming in. And low is way out. So you have to optimize it.
Okay.
Isolate
When
contour.
when we zoom in, then then definitely it will become straight.
Yeah. So you have to
Okay.
zoom out.
Okay. Thank you, sir. Have a
Sure.
nice talk
Where?
with you.
No, that's fine. Dr. Singh, any other questions?
I think Inamuddin wants to ask any question.
Please go ahead.
Inamuddin.
Hello, sir. Good evening, sir.
Hello.
Hello, sir.
Yeah.
Can you hear me?
Yes.
Sir, actually, you discussed biosensor, and I am related to. Hello. Good evening, sir.
Yeah.
Hello.
Go ahead.
Yeah, appreciate.
You discuss biosensor, but
Mhm.
actually DNAs consist a large number of molecules like sugar, phosphates, and nitrogen group. So your attachment is the biomolecules with CNTs, but
Agreed.
how the total DNA attach with the CNTs
So
molecule?
Namuddin is what we are looking at is if you see through the
Ji, sir.
nanopore is coming out is a single strand.
Ji, sir.
Okay. So single strand gives you the charge on the backbone.
Ji,
So you can pull through the nanopore. And what we have is, again, this is a thing is depending on, as you know, GC, AT, like the question was GG, GA. So is going to give you low current or high current.
ji.
And from there, you can distinguish. And remember, I use the word is order of magnitude difference in current gives us distinguishability.
Yes, sir.
Okay. So there are some criterias we have used it. And this low or high interaction strength really gives a distinct signals. I can send you the paper. I have not given all the details from here, but we have looked into every base and every base pair.
Okay, sir, but how we differentiate that this is sugar molecules or DNA?
See, what
On the
happens
basis
is
of
I'm
energy?
not worried about the sugar molecule. My question is, can we have a distinction between ACGT? That's DNA sequencing.
Okay.
So we have the probe molecule is one of the bases, and this hydrogen interaction as it passes through. So what is happening here? Earlier cases, they did not have the probe is passing through. So they had a lot of noise because of the fluctuation. So here the assumption was if I bring in a probe, the hydrogen interaction stabilizes for a short while and then it goes through because we are still pulling through it.
Yes, sir.
Okay. So stabilization by the hydrogen bond interaction was expected to give a little better signal.
Yes, sir.
And they are now, if you look at the literature, people are using it, that approach. It's still expensive. It is still in the lab scale.
Yes, sir.
ठीक है?
Swarnadip Biswas.
Good evening, sir. Sir, it
Hello.
was a very informative. Good evening, sir.
Sure,
Sir,
sure.
it was a very informative talk, sir.
Thank you.
And I have gained many knowledge from you, sir. I'm very much thankful to you for in
Sure.
this regard. Slowly and steadily, I'm learning a little bit from Professor Mohan Verma, sir. And
Wonderful.
the some of the talks are pertinent to the research work that I have taken right now, and
Good.
the one of which is the absorption of nucleobases on graphene surface. Sir, in one
Good.
of your slide, you have shown us that how these
Yeah.
nucleobases are attached or absorbed on the surface of the graphene, sir. So,
Agreed.
what is the size of the graphene that you have taken for that case? Means,
See,
is it
Dr. Vishwas, is size of graphene instance is, as you know, in simulation, defined by your model. So in a sense, you're right, we cannot take 2 by 2 atom because then our molecule is going to cover the surface completely. So what we have is a typical, if I remember, size is called 6 by 6 or 8 by 8. So what happens is here, because you have the periodicity, you don't want these molecules to interact with each other. So you have to have a large supercell of graphene interacting with these molecules, not the standard one.
yes, sir. So it is, sir, 8 by 8.
Approximately, I don't remember exactly, but is a large, yeah.
Yes, sir.
I
Yes,
can
sir.
look into it.
Okay, sir. Thank you, sir. Thank you.
But you have to make sure when you apply the vacuum as well as the boundary condition, because XY you're repeating it, periodicity is there. So these molecules, they have at least 8 to 10 angstrom distance. Otherwise, they're going to have called self-interaction errors. So you have to have such a large cell, so these molecules do not interact.
Yes, sir. I get it. Thank you, sir.
Now we have Mr. Malik.
Naibul
evening, sir.
Malik.
Hello.
Yeah, go ahead, Naib.
Sir, I have performed substitutional doping on SNS2, tin disulfide,
Good,
where sulfur atom replaced by silicon. And before
good.
doping, sir, the DNA RNA interaction with this material, the adsorption energy was very low. But after the replacement, the adsorption energy is into the range. But the
Good.
recovery time and sensitivity did not fall within a desired range. So what should I do, sir? And second thing is I didn't apply the van der Waals interaction. So should I apply it
Which
for this
software package are you using?
siesta?
Siesta. So
Yes.
Naib, you had to have, because you are using this DNA, so they have this pi electron cloud.
Mhm.
Okay. So applying D2 interaction is must.
Okay.
Okay.
D2.
So if you use GGA, so GGA plus D2 and Siesta has D2 interaction. And you
Yes,
can
sir.
see the binding energy is going to change.
Okay.
Okay. Second thing is, when you said you're doing SNS2,
Yes, sir.
and silicon is replacing sulfur or silicon is replacing tin?
No, sir. Sulfur is replaced by silicon. Sulfur atom.
See, this is little different because, you know, silicon is cation
Mhm.
and you are using an ion to replace cation. Is it
Uh,
feasible?
uh, Sir, when I replaced it and after again optimized it, so the atom has been fixed at that place.
No, simulation wise, you can do whatever you want. Okay, it's going to
Mhm.
give you some result. But first thing, Naya, we ask, is it physics wise, does it make sense? When you make a sample, you ask Dr. Vishwas or Iman that how do you make sample? They say they mix it, warm it, the cation is going to go to cation site, not the anion site.
Okay.
Okay, because you know they have called electronegativity differences, and that's why the
Mhm.
ion choose to replace. So my suggestion is, Nayab, go back and then do
Mhm.
silicon substituting tin, not sulfur.
Okay, sir.
Okay, and see what happens.
Yes. Okay, sir.
Because
Sir, when
modeling wise you can do it, but physics wise is not possible.
okay.
I just want to
Sir,
add one.
Is that
one
correct,
line,
Dr.
yeah?
Vishwas?
I just want to add one line, yeah, Professor,
Go
if
ahead,
you allow
Imran, go
me.
ahead.
Yeah, so like what Nayab did, I think so, in experiment, we basically call it two phases material, not a single phase. So I
Oh,
mean,
I see.
yeah. So never, I mean, I hope so that silicon never captured the place of sulfur, never. But
Agreed.
it will exist as a separate phase. So it will become a secondary phases. So basically, you are studying composite, the properties of composite, not a dope material.
But if I understand, Nayab has done the calculation where he replaced sulfur by silicon in calculation.
So, yes, sir. Sulfur
So
atom
that
by
should not be
silicon.
the case. Like Imran said, that should not be the case because silicon doesn't like sulfur, means it's replacing sulfur.
Mhm.
Is a sort of
Mhm.
aggregation or substituting tin. So your model may be correct. You are going to get some answer, but physically that model is not correct model representing the processes.
Okay.
Is that correct, Imran?
Yes, we can't get them experimentally.
Yeah.
Yes, I am agree.
So now we go back and then see what
Mhm.
happens when you use tin site.
Okay, sir. Before I also tried with tin side, but because it is a sandwiched. I mean, the tin layer is in between the sulfur layers.
Agreed.
So when I tried to replace the tin atom, the charge is not go onto the surface, and the interaction, it will not uh, take part into the interaction. Only the upper sulfur layer uh, take more part into the interaction with nucleobases. So the
But
doping.
So
So the
this is
doping.
another issue.
Mhm. Yes.
I think so. You are cutting the surface with the along the along the sulfur. Yeah. So basically, usually in experiment, people cut or the DFT people cut the surface along the metal atoms. I mean, on the top we must have tin, not a sulfur.
Mhm.
I hope
Okay.
Sir Pandey will be
correct.
agree.
That's correct, Imran. That's correct. Yeah. Second thing, Nayab also always remember is your tin sulfur is a tin sulfide. You replace tin by silicon is going to modify the sulfur electronic charge, definitely.
Mhm.
And in turn is going to affect the adsorption properties. So
Ah,
you bring in your biomolecule to sulfur, which is connected to silicon, not the tin.
okay.
Okay, so you choose the target site.
Mhm.
Okay, and you can see if you do have the Bader's charge or electronic distribution, sulfur connected to silicon is going to have different behavior compared to sulfur connected to tin.
Okay. Mhm.
Now the question is this small modification, is it going to influence your adsorption properties? I don't know.
Yes, sir.
Okay, but you have to figure it out first, what
Mhm.
the change is.
Mhm. Okay, sir. Sure, sir. I will try it.
Yeah. Dr. Schwartz, you have one more question? Are you okay?
Sir, for whenever I dope something or create vacancy or do anything, so should I first uh, find out its chemistry?
Agreed. Agreed. And that's chemistry is which Imran and anybody can tell you is the electron negativity or electron affinity of atoms.
Mhm.
So we have called Pauling's scheme.
Mhm.
And from there, which one likes which one.
Ah, okay,
And
sir.
then you have to look at the atomic size also.
Ah, yes, sir.
And that helps you.
Okay. Thank you, sir.
Sure.
Sir, I have another question, sir. Sir, you have
Please.
given insight regarding the layered material. We have to add around 10 Armstrong of vacuum to avoid interactions. For Van der Waal structure, sir, bilayer, how much amount of distance to be taken for that purpose?
See, Van der Waal reaction, if you have bilayer, is of course for a highly accurate, Keshav can tell you. As Keshav, you use about 15 to 20 angstrom vacuum. He's not there. Neha, do you remember if you're there? So Dr. Schwarz, about 10 to 15 angstrom is good enough.
Okay, sir. Okay, sir. And
Yes.
between the two layers, sir.
Oh, Neha is here. Neha can tell you.
Okay.
Yes, sir, 10 to 15 angstrom is
It's
good.
good enough.
Yeah,
Yeah,
it's
good.
good enough.
And
There
for
should not be
the two
interaction between the
layers,
two layers. Nah, it's sufficient that it's
Yeah.
fine if we use 10 to 15 angstrom. That is good. Yeah.
okay, ma'am. Thank you.
So Dr. Schwarz, at my age, I don't do these calculations. I rely on Neha and Keshav and others.
Yes, sir. Yes. Okay.
Good. So is it 10 o'clock? Dr. Singh, I had to go for my.
Thank you, sir. Thank you, sir. Just one
Thank you
request,
very
sir.
much.
Just one request, sir. Let it be. I mean, it is recorded. Just you have to save it, sir.
I'll save it and then we'll send it to you. And also I'll send you the slides, okay?
Yes, sir. Yes, sir.
You can distribute it.
Thank you, sir. Thank you for
Take
this
care.
wonderful.
Thank you, Mohan.
presentation,
Thank you,
sir.
Neha.
Okay.
See you soon.
Sir.
Thank you,
Thank you,
Keshav.
sir. Thank you so much.
Yeah, take care.